great pleasure to introduce Michele, or Mickey as he's known to us. So Mickey trained as a molecular biologist, stroke biochemist at the University of Perugia and the University of Padua in Italy, and then moved to Nick Turner's group to do his PhD working on discovery engineering and the application of cytochrome P450s. And during that PhD, spent time in John Woodley's lab looking at the scale up of the processing of that. When he graduated from his PhD, he joined Sabine Flitch's group to work further on oxidases for a short postdoc before GSK recruited him in February 2019 to work on a European Union era co-biotech project called BioDimet. And Mickey's work has been focusing on the industrializability of cofactor recycling, and he's going to talk to you today about his work that's been done in terms of industrializing ATP recycling. So I'll hand over now to you, Mickey.
Yeah, thanks, Mari. So hopefully you can see my slides.
Yep.
You'll be hearing about ATP recycling, as Mari said, and thanks for the introduction, and thanks to the organizer for putting us together to be able to share the science during this time. ATP-dependent enzymes are widely distributed in nature, and they allow very interesting and challenging reactions that I'm showing here on the left. There are some issues associated with these enzymes when stoichiometric ATP is used, and these drawbacks are related to the process costs, which is introduced by using a stoichiometric factor, substrate and product inhibition, and reaction equilibrium limitation. So if we want to do these interesting reactions in industry, we need scalable technology for ATP recycling. So there are well-established ATP recycling systems, but they typically allow for ATP regeneration from ADP, whereas some applications, such for example carboxylic acid reductases or amide bond synthetases, would require recycling of ATP from AMP. And then there are some issues associated with the phosphate donor cost, stability, and availability for these more mature ATP recycling enzymes. On the other hand, polyphosphate kinases, PPKs from now on, use cheap and stable phosphate donor, which is a chain of phosphate units, polyphosphate. And they also allow ATP regeneration from AMP. So generally speaking, there are two major classes of polyphosphate kinases, PPK1 and PPK2, and the latter are typically used for ATP synthesis and recycling. Here on this scheme, you can also see that PPK2 enzymes are classified in three additional classes according to the preferred catalyzed reaction. So we have PPK2 for the synthesis of ADP, PPK2 for the synthesis of ATP, and then the bifunctional PPK2 class 3 that can go all the way from AMP to ATP. So it has been recently reported that actually, regardless of the class of PPK2 enzyme we are considering, this biocatalyst can synthesize ATP from AMP, even if we are considering a PPK2 class 2 or PPK2 class 1. So this was a very interesting finding that makes these enzymes even more interesting. So the aim of this work was, first of all, to acquire and investigate PPK2 enzymes across different classes, and we're trying to plug this gap in ATP recycling whereby there aren't well-established technologies for ATP recycling from AMP. And the aim of this work was to find a single enzyme to do this job, which could be used as a crude extract, so as a lyophilized cell-free extract. And obviously, being in industry, we are interested in investigating the scalability of this system. So to start with, we have assembled a small panel of 11 polyphosphate kinases, mainly reported enzymes, to which we have added some other enzymes from extremophiles. And we screen them as a lyophilized cell-free extract against AMP. So here on the x-axis, you can see time. On the y-axis, the percentage of AMP, ADP, and ATP indicated with different colors. And as you can see, if you just follow the red here, there were quite a number of enzymes capable of synthesizing ATP from AMP, regardless of the class we are considering. So I'm indicating also the class for each of these enzymes on top, close to the name. So in particular, AJPAP, RMPPK, RPPPK and the RPPK were capable of converting about 30-40% AMP to ATP within 15 minutes. So this first set of experiments showed us that we were capable of polyp-driven AMP to ATP synthesis using various enzymes as a crude extract. Subsequent work focused on the intensification of ATP synthesis and led to the selection of AJPAP, this enzyme up here, top left, for subsequent work. So we then moved from ATP synthesis which is what we have been investigating with these experiments, to ATP recycling. So we wanted to test these enzymes in a real life reaction. We defined a model system to do that, and we selected the carboxylate reduction to aldehyde catalyzed by Cas. These are quite interesting enzymes that have some structural and mechanistic features that allow for reduction of carboxylic acid to the aldehyde with no over-reduction to the alcohol. And in doing so, they require NADPH for reducing equivalence and ATP. So we screened an in-house panel of carboxylic acid reductases with a panel of substrates, and in the end we decided to go with this CA33 and 4-methoxybenzoic acid, obviously implementing AJPAP ATP recycling powered by polyphosphate. In applying this carboxylic acid reductase system, again using crude enzymes, we decided to adopt a biphasic reaction mixture comprising a 50% toluene, because in order to mitigate aldehyde over reduction to the alcohol due to enzymes from the host organism. But we rapidly realized that by using this reaction system, the poor mixing was probably limiting the performance. The poor mixing in a small scale setup was probably limiting the overall performance of the system. So we decided to move, as I'm trying to show here, into reactors with a more controlled steering and pH control to try to exploit all the advantages of these systems. We performed a series of reactions on a 30 ml scale, and then we transferred these conditions on a 400 ml scale. So here on the left on this table, you can see how an overview of the gram scale synthesis of 4-methoxybenzaldehyde. So we charged our reactor with about 5 grams of substrates, and a process lasting about 44 hours. We got multigram amounts of 4-methoxybenzaldehyde. So on the one hand, these results were quite exciting, but because this is one of the few, if not the only report where multigram amounts of aldehyde can be made with a car in a fully in vitro system. So there are no old cells, and we are using a crude enzyme preparation. We are not using purified proteins. So there is little about this in the literature. But on the other hand, there were some red flags. So first of all, honestly, running this process was a nightmare because it required multiple doses of reaction components over 44 hours, and the ATP recycling loading here. The ATP recycling enzyme loading here was a little bit too high. We were aiming at something on a single digit, so lower than 5% weight to start with. In addition to this, we carried out some follow-up experiments that highlighted some stability issues associated with AJPAP, plus the significant support of host background activities for ATP recycling, and we were not happy with that. So then that simply meant that we needed to find a better PPK. We need to find better enzyme, both in terms of activity without assistance from host proteins, and also finding a better enzyme in terms of stability. And as the overall aim of this was to try and find an enzyme for ATP recycling from AMP, this time we decided to focus our panel on PPK2 class 3 enzyme in order to increase the chances of getting an enzyme to do the job we wanted to do. So we started our investigation with PPK2 related proteins, and we exploited the differential clustering of PPK2 sequences belonging to different classes to retrieve PPK2-3 enzymes. Once obtained these sequences, data reduction followed to remove short sequences to long sequences not well annotated, and finally genes were obtained from bacteria, archaea, and metagenomes, to which we've added some previously reported PPK2 class 3 enzymes to carry out a comprehensive comparisons between these enzymes. So, as I said, the one of the aims of this panel acquisition was to find an enzyme that could run ATP recycling unsupported, so without enzymes coming from the lysate. So the primary screening was carried out with purified proteins, so enzymes were produced in E. coli, and high throughput protein purification performed. So as you can see here, where I'm showing reaction time on the x-axis, so 15 minutes or overnight incubation, and in the y-axis, I'm showing percentage ATP. Looking at the right part of the graph, where we are looking at data after overnight incubation, you can see that there was quite a significant fraction of enzymes that we picked directly from nature that were capable of forming ATP starting from AMP. Within this, there was 16% of enzymes, of which 13 were novel enzymes that could convert more than 50% AMP to ATP within 15 minutes. We carried out some follow-up experiments at reduced purified protein loading in order to be able to select the front runners, basically. For the secondary screening, we focused on the activity and the stability of these enzymes as lyophilized cell-free extracts. So we were moving towards our envisioned process, basically. So here in A, you can see the percentage ATP conversion of the 8 selected enzymes at different poly-P loading, at different polyphosphate loadings, and as you can notice from the shades of gray, there is quite a sharp decrease in the ATP synthetic activity at increased polyphosphate loading. So as we are trying to intensify these reactions, we are losing activity. But there were two enzymes, PPK11 and PPK12, that maintained a good fraction of their initial activity, even at 400 millimolar polyphosphate. On the bottom here in B, we have investigated stability and versatility of these enzymes. So the biocatalysts were incubated at 30 degrees from 2 to 48 hours, and then reaction were run at different conditions, so that you can see here on top. Here I'm showing data for enzymes pre-incubated for 24 hours. So again, here you can see that PPK12 was quite stable enzyme, versatile biocatalysts under different conditions, and this feature combined with the high polyphosphate tolerance of the enzyme led us to select this biocatalyst for subsequent work. So again, we moved from ATP synthesis to ATP recycling. So we moved again to our carboxylic acid reductase system. And here you can see a comparison between processes run with different ATP recycling enzymes and at different loadings. So we have AJPAP at 1 in blue or 10 megaml loading in red, and then we have PPK12 at 1 megaml or 10 megaml. So by just using 1 megaml of PPK12, we could form more than 50 millimolar products, whereas less than 45 millimolar products were formed with 10-fold more AJPAP. So this experiment underline the fact that we really found a promising and good biocatalyst for scale up. So we moved again to we run another 400 ml on another carboxylate reduction to aldehyde on a 400 ml scale, and in this with PPK12, and in this case, such an extensive optimization was not required for the PPK12 implementation, so we could run everything in batch, and this led to a much simpler process to run compared to the AJPAP process. And as you can see here, the process time was 22 hours against the 44 hours process time with AJPAP, and the implementation of a more active and stable PPK2 enzyme resulted in a lowered ATP recycling loading. So we got more than a 4-fold reduction in the ATP recycling loading, approaching the numbers, approaching the process metrics that we set at the beginning. So to conclude this talk, we have seen the initial selection of an enzyme based on a small panel, and the scale-up revealed some limitation associated with the enzyme. We went back to nature. We assembled a panel of PPK2 class 3 enzymes, and we've added more than 50 novel enzymes that can be used for ATP recycling. And in seeking a more active enzyme, we applied a tiered screening approach where we looked at polyphosphate tolerance, stability, versatility, and finally we found an enzyme that powered a multigram scale cell-free aldehyde synthesis at significantly lower loadings compared to the process implementing AJPAP. So we think that this is really a significant step in industrializing ATP recycling, even though there are some things that we probably need to look at, additional demos, directed evolution, and other improvements. So for this, I thank the EU for funding, and I thank all my colleagues at GSK that contributed to this work. I'm happy to accept questions.
Hey, thank you, Mickey. Great talk. It looks like we have a couple questions in the Q&A. We have time for a few questions. Just a reminder, it is different than the chat function. So if you're putting questions in the chat, just make sure to move them over to the Q&A. Also, if you're watching on the YouTube live stream, we do have someone taking questions from there and bringing them to the Zoom. So you can ask questions. Anthony, I'll pass it over to you.
Hey, Mickey, great talk. So first question, did you investigate immobilization as a way to reduce host background activity?
Nope, we've never attempted immobilization. We could have, we could have, for example, these proteins are all histidine tagged, so we could have used technologies that allow that. But no, so our aim ultimately would be to have such a good enzyme that we reduce so much the loading that host background activities are not a problem anymore.
Okay, so a question from Bernard Hauer. Can you bring your results in perspective with similar reactions based on electrochemistry?
Sorry?
Sorry, can you bring your results into perspective compared with similar reactions based on electrochemistry?
I wouldn't know how to do that. No, I wouldn't be able to give a perspective based on this.
Okay, that's no problem. Okay, so we have a question from Tristan Duron. Could you comment on the benefits of performing ATP-dependent enzymatic transformations in cell lysate versus a whole cell bioconversion? Brackets,
yeah,
in the way
so
that go on, Mickey.
Yeah, so this is actually a very good question, because we obviously in industrial biocatalysis we are trying to use the crudest biocatalyst form possible. So there are examples with old cells, but the issue with old cells is so by using old cells, this sort of in vivo biocatalysis, we might encounter problems related to mass transfer problems, for example, or toxicity of the material you are using to the cell itself. So it can damage the metabolism that you're trying to use to run the reactions. And what we find also is that typically old cell reactions are the downstream is a little bit more messy. So our favored biocatalyst formulation is typically lyophilized cell-free extracts, because it removes a little bit of burden on the downstream, and we can uncouple protein production from its utilization. So yeah, that's why we prefer lysates to old cells.
Okay, maybe just time for one more question, because there's quite a lot of questions going through. So from Scott France, did you see any variation in performance with different poly-P sources / chain lengths?
It's something that I haven't investigated directly, but at the onset of this work, we have also used, so for this we've been using sodium hexametaphosphate, so it's quite we're not using a defined chain length of polyphosphate, but before doing so we were using another form of Graham salt, and the performance was not as good as with sodium hexametaphosphate. Yeah, so there is this trend.